So, the topic to be discussed today is haptens and antibody. Haptens is a word derived from a Greek word which is known as haptein. That means to fashion. To fashion means a compound which doesn't have a particular property, but it can develop this particular property when this particular compound is complex with one or more uh, larger carrier molecules, and that larger carrier molecule should be a protein or complex protein. So when this molecule is not that that molecule should be protein, it can be a synthetic organic molecule or can be an organic molecule derived from plant or microorganism, can show their antigenic behavior. Mostly they can show their immunogenicity when they will bound with a larger carrier protein molecule. So one of the example of hapten is penicillin. So you people know that penicillin has been discovered by Alexander Fleming from the fungus Penicillium notatum, and from its discovery, this particular molecule, beta-lactamase, uh, this particular molecule is known as an antibiotic or used as an antibiotic. But when this particular molecule is administered orally, so some individual may develop adverse reaction or allergic reactions. So how this allergic reaction is happening there is a type of immunogenic response or immune response. And this particular immune response has been developed by penicillin when it combined with certain serum proteins of the sensitive individuals. Now the complex protein will result in immunogenic property and activates the lymphocytes and initiates severe and sometimes fatal allergic reaction. So penicillin can be considered as a hapten. There are two types of hapten. One is a complex type, the simple type. So the simple type of haptens are monovalent, means they have one binding site, single binding site. And when they complex with specific antibody, so no precipitation is there, maybe agglutination or flocculation reaction. Example, penicillin, 2,4-dinitrophenol, etc. But in case of the other haptens, that is known as a complex hapten, they have multiple binding sites, means they are multivalent. And when they complex with specific antibody, there will be precipitation. One of the example of complex hapten is dextran 1. So there are some examples of hapten except penicillin and dextran 1. So aniline, a purely organic compound, benzoic acid, can also function as hapten, and these are the first research hapten. Then the hydrolazine generally function as a vasodilator prescribed for the patient suffering from high blood pressure to lower their high blood pressure. It can also function as a hapten. The fluorescein dye, fluorescein isothiocyanate, vitamin biotin, and then decoxygenin is a steroid derived from plants mostly the digitalis plants, digitalis purpurea, digitalis orientalis, digitalis lantana, lanata. And this is a species of foxglove plant. From their flowers and leaves, we can collect the steroid, and this steroid can be used, can act as a hapten along with 2,4-dinitrophenol. Now, as because the haptens are showing antigenic behavior, so what are the similarities between hapten and antigen? So both are antigenic and present on external cellular surface of microbial pathogen or maybe the other agents. They form a part of the defense mechanism system between antigen and an antibody, and they have the ability to bind to the antibody via weak interactions like salt bridge or ionic interaction, hydrogen bonding, dipolar interaction, hydrophobic interactions, van der Waals interactions like that. And this similar phenomena we can also get in case of antigens. Now, what are the dissimilarities between antigens and haptens? So, antigens are the foreign body which has the ability to trigger the host immune response, or host immune system, to produce an immune reaction by binding with an antibody. The haptens, they are actually, we can say they are incomplete antigen. And solely or individually, they cannot show immunogenicity. So antigen directly bind with the antibody. And after binding with the antibody, they can initiate an immune reaction. But haptens, they binds to an antibody but doesn't have the ability to trigger the host immune response or host immune system to produce immune reaction. So antigens have both antigenicity as well as immunogenicity, but the haptens have only the immunogenic property or immunogenicity. They don't have the antigenicity. This is the difference between antigens and antibody. Sorry, antigens and haptens. Now, what are antibody? So, if I want to define antibody, so antibodies are specialized glycoproteins produced from the activated B cells or the plasma cells in response to an antigen and triggered its production and also initiates acquired immunity of the host cell. So antibodies are glycoproteins, mostly glycoproteins. And what are the proteins? What are the glycoproteins? They are serum glycoproteins. So in your blood, you can find different proteins or glycoproteins there. So which are the proteins that can function as an antibody? So if you isolate serum from a human being and then you perform electrophoretic reaction, SDS page, sodium to decyl sulfate polyacrylamide gel electrophoresis, because this particular technique is used for the separation of different proteins. So you can see that proteins will migrate through this when they are placed in an electric field from positive to negative, or else negative to positive, negative to positive, or not from negative positive to negative, it's negative to positive, because the because SDS is imparting negative charge toward the polypeptide surface. So here you can see that the albumins, they will travel more. So we can have 4 to 5 different fractions. And the protein that has migrated more is albumin. And then we can found 3 to 4 different bands of, and that are the globulin protein. We can find 3 different fractions of globulin protein, alpha, beta, and gamma. There are 2 different types of alpha, alpha 1, alpha 2. And it is the gamma fraction that is actually showing immune response. So the serum protein that are migrating less towards the positive electrode when they are placed in an electric field in SDS phase and showing immune response is known as antibody. So antibodies are associated with the gamma globulin. Now antibodies can also be termed as immunoglobulin because by chemical nature they are globulin, gamma globulin, and their property, they shows the property of immunogenicity. That is why antibodies can also be termed as immunoglobulin. So these immunoglobulins are glycoproteins, where the protein content is 82 to 96%, whereas the sugar content or carbohydrate content is 2 to 14%. And they are actually the humoral branch of immune response, not the cell mediated, it's an humoral branch of immune response. And out of the total protein, serum protein, 20 to 25% of the total serum protein belongs to immunoglobulin. So the first use of the term antibody occurred in the text of Paul Ehrlich. Then Garland Morris Edelman, who shared the Nobel Prize in 1972 in physiology and medicine for his work with Rodney Robert Potter on the immune system. So Edelman's Nobel Prize winning research concerned uh, the discovery of the structure of antibody molecules. Why is a structure of antibody molecules? The first evidence that antibodies uh, were, were contained uh, in particular serum protein uh, fractions from a classic experiment performed by Tiselish and Cabot in 1939. Now, primary role of antigen antibody is the recognition of antigen and then show biological activities related to immune response as well as complement activation. So these are the functions of antibody, and there are 5 classes of antibody: IgG, IgA, IgM, IgT, and IgE. And the number of antigen binding site of an antibody is called the valency of the antibody. And most of the cases, human antibodies are bivalent because they have 2 antigen binding site. Now, the structure of antibody. So, antibodies are Y-shaped, heterodimers. Means there are 2 different type of protein composed of 4 polypeptide chains. So, 2 identical yellow chains, they are showing the light chains, means low molecular weight, a polypeptide chain of uh, 25 kD each, and 2 identical heavy chains, each having molecular weight of 50 kD or more. So all these 4 heavy and light chains are bound to each other by disulfide bridge bond, and as well as non-covalent interactions such as salt linkages, such as ionic interactions, ion dipole interaction, hydrogen bonding, uh, van der Waals forces, hydrophobic bonds interactions. That. So the covalent interaction here is a disulfide bridge bond, and the other interactions are very weak interactions. So all the chains have 2 ends. One is a carboxyl end, C terminus, and other one is a amino end, that is called the N terminal. And mostly this N terminal end is in a variable chain, uh, variable part, and the C terminal is a constant part. And this immunoglobulin, I said that these are tetrapeptide because 4 polypeptide chains are there. And this intrachain along with this interchain disulfide bridge bond, we can also found intrachain disulfide bridge bond in between the heavy chain mostly, as well as in between, as well as in the light chain, and they are forming some domain. So amino terminal of each chain is a variable region, while the carboxy terminal end is one or more constant region. Now, if we use the proteolytic enzyme, papain and pepsin, so these proteolytic enzymes yields two different fragments. One is known as a Fab fragment, and the other one is a FC fragment. So we can get two Fab fragments and one FC fragment after digestion by pepsin or papain. So here you can see enzymatic digestions generate various fragments. So mostly this proteolytic enzymes function as the hinge region or the flexible region of the antibody here. So they cleave here. So the disulfide bridge bond of the hinge region, and they will generate two Fab fragments having molecular weight nearly 45 kD each, and one FC fragment molecular weight 50 kD. In case of the digestion performed by pepsin, it's also similar. They are also generating two Fab fragment and one FC fragment. The smaller fragments are the Fab fragment, and the many smaller fragments also. So what is Fab region or Fab fragment? So Fab region is the top part of the Y, consists of two antigen binding sites. And these regions are composed of both the variable as well as the constant region. So molecular weight, as I said, 45 kD. In case of FC region, this is the stem or lower part of the Y-shaped antibody monomer, molecular weight 50 kD. And this is very important because immunological reactions, this particular region shows immunological reactions, and it's composed of only the constant region, no variable region present in the FC fragment. And this particular fragment interacts with the cell surface receptor called by the FC receptor and some proteins of the complement system. So this property allows antibody to activate the immune system or the immune response of the body. Now we have got two different chains. We have two different chains. So two chains of more amino acid chains. So these are called the heavy chains, and two are light chains. So heavy chains have 5 classes, and these 5 classes are designated by Greek letters, and they are structurally and antigenically distinct. So these 5 classes are IgG, IgA, IgM, IgT, and IgE. And this class, they have been classed based on the amino acid sequence of each. In case of the light chains, there are 2 types, and these polypeptides are called as Benz-Jones proteins. Latter, uh, Korngold and Lapari termed as kappa and lambda chain, kappa and lambda protein. So in human, L chains contain 60% kappa and 40% lambda type. So both light chains of antibody molecule should be similar type, means either they are formed by kappa or else lambda. So you cannot find that both the chains belongs to kappa and lambda. So if it is kappa, means both the chains belongs to kappa. If it is lambda, both the chains belongs to lambda. So you cannot find never both. And here you can see this is the H chain and the L chain domain. So further it can be subfolded into some subdomains. Here you can see is some folding there. So 60 amino acid, they contain 60 amino acid. So each and every domain they are folded like that. They are forming a loop-like structure, and each and every loop contains 60 amino acid. They have the intra-chain disulfide bridge bond. So cysteine-rich amino acid is there, or chain is there. So sulfur-containing amino acids is forming the intra-chain disulfide bond. In case of light chain, you can find two region. One is a variable region that is marked as VL, and the other one is a constant region that is marked as CL. In case of heavy chain, you can find 4 to 5 regions. One variable, this is constant, only one variable as VH. You can find 3 to 4 constant region in the form of CH1, CH2, CH3, and CH4. So it varies from 3 to 4. Now, what are the types of heavy chains in each immunoglobulin class? So you can find 5 different chains, 5 different immunoglobulin class. Mostly you can find in IgG is gamma heavy chain, IgA alpha, IgM mu, IgG delta, and IgE is epsilon. So these are the Greek words or chains that can be expressed in Greek word. Now the variable and the constant region comprises of two regions. One is known as a variable region and the constant region. Both the H and L chains you can find these two regions. In case of the light chain, we can find one each, and in case of heavy chain, we can find one variable and 3 to 4 constant region. So the variable region, so first 110 amino acid residue near from the amino terminal end, the N terminal end, and of both L and H chain constitute variable regions in the form of VL or VH. So they represent the antigen binding site of the antibody, this variable region, variable domain. Some hypervariable regions are also there. Some zones are these are termed as hotspots in the variable region, so relatively higher variability in amino acid sequences. So this is known as complementarity determining regions or CDRs. Uh, from the antigen binding site, you can find this 3 to 4 hotspots in, sorry, 3 hotspots from the antigen binding site. You can find the 3 hotspots in L and 4 hotspots in H. Now, the site on the hypervariable region that makes actual contact with the epitope of an antigen is known as antigen binding site or paratope. So, actually, the antigen binding site of an antibody is known as paratope. That is there in the that is there in the variable region of an antibody. Now, what is epitope? We have already discussed about epitope. Epitope is the antigenic property. So, the small site present on the surface of an antigen or present on the antigen, which is recognized by the paratope of an antibody, the antigen binding site of the variable region of an antibody. Usually between 1 to 6 sugars or amino acids on the surface of an antigen present there, and they are actually composing the epitope site of an antigen. Next one is the hinge region. So this hinge region belongs to the heavy chain, so gamma, alpha, delta. So you can never find hinge region in mu and epsilon chain. Only you can find in this region alpha, gamma, and delta. The junction formed between the constant region of the heavy chain CH1 and CH constant domain of the heavy chain CH1 and CH2. So this region is a flexible region, is known as the hinge region, and this particular region is rich in proline and cysteine. So you can find intra-chain disulfide bridge bond here. So flexible amino acids stretch in the central part of the heavy chains of the immunoglobulin G and immunoglobulin A classes, which links these two chains by disulfide here, by this disulfide bridge bond. They are sensitive to this particular region or location is sensitive to proteolytic enzymes like pepsin and pain. So enzymatic digestion of protein of amino acid of antibody occurs in this particular region or location. So you cannot find this region in mu and external chain of antibody. Now constant region. So the remaining part of the immunoglobulin molecule at the stem part of the lower part of the Y-shaped immunoglobulin molecule is known as the constant region. The length, it contains 104 amino acid for the light chain and 330 amino acid for gamma, alpha and delta heavy chain and 440 amino acid for mu and epsilon heavy chain. So you can see here a variety in amino acid uh, chain length, amino acid number that is the length of the polypeptide in the constant region, and it is different in case of light chain and heavy chain, and in case of heavy chain also the three different chains alpha, gamma and delta have 330 amino acids, whereas mu and epsilon have 440 amino acids. So amino acid sequence of constant region shows a uniform pattern, and a single antibody has two identical H chains and two identical L chains. So this is all about today. Next day we will start with the classification of